seconds, and then I will start. Welcome to the ICC Webinar series. Our topic today is sourdough, a tool to improve flavor, sustainability, and digestibility. First of all, I'd like to tell you that our moderator will be Alfred Marr. After giving a short introduction about ICC, I'll hand over to Alfred Marr. If I can get the next slide, I will give you some information about ICC. ICC was established in 1955. The first name was International Association for Cereal Chemistry. Now is International Association for Cereal Science and Technology. ICC is an apolitical international network of cereal scientists and technologists, and we are active all over the world. It is one of the most well-known international organizations in cereal science and technology, dedicated to international cooperation, the dissemination of knowledge, and improvement in safety and quality of cereal-based foods. As I said, today's topic is sourdough, a tool to improve flavor, sustainability, and digestibility. This webinar today is organized by ICC and Boku. We always had good collaboration with University of Natural Resources and Life Sciences. So we have two speakers from Boku today. And with the moderator of Alfred, and now I would like to hand over to. I would also like to thank Alfred because in ICC we always had very good support from Alfred Marr. So and with my thanks, I will hand over to Alfred, and I will just mention the names of the first speakers, but Alfred will give more information. Lisa Cole and Vera Frabeck from Boku University. Thank you very much again for participating our webinars, and now Alfred Marr will moderate the rest of the webinar session today. Thank you very much for participating.
Dear Hamid, thanks very much for the kind introduction. Thank you very much. Dear cereal scientists and technologists, I welcome all attendees of our today's webinar, Sourdough. My name is Alfred Marr, as Professor Hamid Köksal pointed out. Beside my function as lecturer for cereal technology on the University of Natural Resources and Life Sciences in Vienna, abbreviated called Boku for the German word University of Bodenkultur. I am also the president of ICC Austria, and it's a really honor to present and moderate the sourdough webinar today for all of you. First of all, let me point out some technical information. The two speeches of Vera Fraberger and Lisa Kahl, I will present and introduce them to you a few minutes later. I give you now the information that the two presentations will follow without a break in between. Very important, we have polls in the presentations. You will see just one at the beginning very soon. Please engage in our polls throughout the time it will take, so you have enough time, but please engage in filling out the points of our poll. Further on, you are invited to post question in the so-called question box. You see it on the right side on this slide where you find it, and please enter the question in the green field you see there, and please address it to speaker number 1 or 2, and you also can use, of course, the names of Vera for number 1 and Lisa for number 2 if you prefer it. At the end of the webinar, we will try to answer all the questions you post in, but at this moment, please, I ask you for understanding. If there is not enough time, you will get answers not within the time of the webinar, but later on per email. Okay, so let me introduce the two speakers. Now I'm very glad to introduce and let me start with the first speech. The first presentation is held by Vera Fraberger. Vera Fraberger worked in the PhD thesis, especially for sourdough, and some of the results she achieved you will hear in this presentation today. And we are very proud that she is one of the young scientists graduated the PhD at BOKU. Also, the second speaker will be Lisa Karl. Also, she graduated PhD on BOKU, and also she was working within her PhD thesis about sourdough, but in a very different topic of sourdough. Rather about tolerability of the components and so on, you will hear. So we really can be very curious about the presentations, because we will hear really actual results. Some of them are also published and some are not. Okay. So before Vera starts the first presentation, sustainability improvement by sourdough processing, I ask Marcella to show the first poll. And as you must know, I am in the sourdough topic for really nearly 4 decades, and the importance of sourdough changed within this time frame of 4 decades. And now I ask you, what is the most important benefit of sourdough in your opinion, of course? Is it improved process quality of dough? Is it improved sensory quality, for example, flavor and texture? Is it extended shelf life, improved tolerability and digestibility? Or is it all above mentioned? Please fill in your answers, post your poll answers. Thank you. Okay, thank you very much. We have the result of the poll. 80% voted for all above mentioned, 17% for improved sensory quality, and 3% for improved tolerability and digestibility. Now We are very curious what will be the outcome of the presentation, and I ask Vera Frauberger for the first speech, sustainability improvement of sourdough.
Yeah, thank you very much, Alfred, for your nice introduction. I hope you can see my slide already. If not, then please tell me. But it should be fine. Yeah, so I also like to welcome all of you. And yeah, today I'm going to talk about if there's the potential of sourdough processing to improve sustainability. And yeah, we will see. And first of all, I'd also like to ask you a poll question. So what's your opinion? So yeah, what do you think? Can sourdough processing contribute to sustainability of bakery products or not? Yeah, if you're certain that it does, then please vote for yes. If you're certain no, not at all, then please vote for no. Maybe some of you are still not certain about the answer, so then please vote for still irresolute. So yeah, if you think that maybe less bakery products are just thrown away, you can check yes. If you think no, it doesn't matter, and no. So yeah, I would like to ask you for a vote. So, and we see that most of you already think that sourdough processing contributes to sustainability of bakery processes. That's of bakery products. That's nice to hear. Some of you are still not certain. Maybe I can change your opinion during the presentation. Yeah, we will see. So back to the presentation. First of all, an overview about what will happen in the next approximately 20 minutes. So first of all, I'd like to give you a short introduction to Boko, where Lisa and I am currently postdocs. Then I will give you a short introduction on sourdough. What is sourdough? How is the microbiota of sourdough? And how can it influence the end quality of the bakery product? And then I will go into detail of deterioration and spoilage of the bakery products, especially by microorganisms, namely by fungi and bacilli. And last but not least, I will finish with a short conclusion. So yeah, you already heard the abbreviation of our university is just BOKO. The full name is University of Natural Resources and Life Sciences, Vienna. So it's already in the name of the university that the university is mainly located in Vienna, so in the capital city of Austria. But we also have a location in Lower Austria, in Tulln. So Lower Austria surrounds Vienna, and a few kilometers outside of Vienna in Tulln, we also have a quite nice location of Boko. I am currently working at the Department of Food Science and Technology, the Institute of Food Science, which is located in Vienna in the so-called 19th district, which is from my point of view, the most beautiful district in Vienna. You see these beautiful pictures of the beautiful vineyards in the 19 districts. And when you're up there and you have a nice day, you have like a really beautiful view over Vienna. So if you ever come and visit Vienna, it's like a must to go there and walk through the vineyards. But back to the institute. Within our institute, we have several working groups who are closely connected together. For example, we have the Food Quality Assurance Group, the Food Sensory Science Group, Food Chemistry Laboratory and Food Physics Laboratory. And I'm currently working at the Food Microbiology and Hygiene Laboratory under the supervision of our head of the institute, Konrad Domig. And yeah, within the group of food microbiology and hygiene, I am focusing, or my research is focusing on sourdough and on the impact of the microorganisms present in sourdough on health-related issues and functional properties. So yeah, let's start with the question, what is sourdough actually? So sourdough is quite a traditional product as it dates back to approximately 5000 BC. And already then the sourdough was used as a leavening and acidifying agent. However, during the Industrial Revolution and due to the current or industrial production of baker's yeast, the sourdough production somehow fell into oblivion. Still, a few bakeries kept using sourdough as it had a diverse range of benefits on the end product. And so, especially within the last years, the sourdough processing regained a momentum. And yeah, especially in the last year, I guess many people started their own sourdough productions at home, now bake their own bread at home. And yeah, the quality benefits are diverse. These benefits or advantages regarding the quality are mainly due to the microbiota of the sourdough. The microbiota of sourdough mainly consists of lactic acid bacteria and yeasts, whereas the lactic acid bacteria are present in higher counts with more than 10 to the power of 8 colony forming units per gram, and yeasts are present in lower numbers with approximately 10 to the power of 8 colony forming units per gram. So they are mostly present at ratios of lab to yeasts of 100 to 1 to 100 to 10. What we also saw in our tests on Austrian traditional sourdoughs. Regarding the lactic acid bacteria, a broad range of species has already been isolated, especially of the genera Lactobacillus, Pediococcus, Weissella, Enterococcus, Leuconostoc, Lactococcus, and Streptococcus, with species of the genus Lactobacillus actually dominating. However, since approximately 2020, there was a big change because the genus Lactobacillus comprised of 261 species until March 2020. So that's really a huge amount of species. And these species were kind of extremely diverse at the phenotypic, ecological, and the genotypic level. And so the taxonomy of the Lactobacillus was reclassified. And now it was changed to 25 new genera with the Lactobacillus genus still present. So now we have to change that not Lactobacillus species are dominating the microbiota of sourdough, but a diverse range of genera is present, like, for example, Fructilactobacillus, Furfurilactobacillus, Lacticaseibacillus, Lactiplantibacillus, Campanilactobacillus, Levilactobacillus, just to name a few. As key organism of traditional sourdough processing is Fructilactobacillus sanfranciscensis seen, as it was already isolated from a broad range of traditional sourdoughs all over the world. Regarding the yeasts, species frequently isolated were from the genera Saccharomyces, Candida, Kazakhstania, Pichia, Torulaspora, and Vickerhammeromyces, where at the species Saccharomyces cerevisiae is assumed to be the dominating yeast. However, there is still a debate going on because most research on sourdough focuses on the lactic acid bacteria. And so there might be the possibility that the occurrence of Saccharomyces cerevisiae is overestimated and that maybe other species are underrepresented. That's still a research that should go further on. And the microbiota of sourdough is actually responsible for the many advantages sourdough processing has on the end product. And for example, lactic acid bacteria and yeasts and the metabolites they produce have health promoting effects. For example, several strains of lactic acid bacteria can produce GABA, which is a health promoting metabolite. My colleague Lisa will then further discuss or focus her presentation on the digestibility and tolerability. So also in this case, the microorganisms might have an influence. Nutrient bioavailability is also increased as due to the decrease of the pH, the phytic acid is decreased and so minerals can be better available. And today I am then going to focus more or less on the shelf life of bakery products, of maybe an extended shelf life due to metabolites produced by lactic acid bacteria and yeasts. And this is also somehow connected indeed to safety of the baking products and also somehow flavor and texture. A picture I guess most of you have already seen and are often shocked about is that tons of bread are thrown away every day, every year. And alone in Austria, approximately 200,000 tons of bread are thrown away per year. So that's a huge amount, and that's actually an issue that has to be talked about. Why is bread or is bakery product just thrown away? On the one hand, because the bread or the staling of the bread has begun, so the crust is or was getting soft, the crumb is getting firm, and therefore the texture and the flavor, has decreased. On the other hand, indeed, microorganisms can spoil the product, and therefore it has to be thrown away. In the case of spoilage of bakery products, especially bacilli play a role, but the most common spoilage microorganisms are fungi. In the case of bacilli, the endospores of the bacilli can contaminate already the raw materials, or they are within the environment, so on the baking equipment, but they are also surrounding us within the air, so they can already contaminate the raw materials and further the bread. And that's also in the case of fungal spores. And in the case of the endospores of the bacilli, unfortunately, endospores are quite heat resistant, so they can survive the baking process, and they even kind of like the higher temperature, so the endospores can again germinate and then spoil the bread after baking. And this is kind of obvious or visible after already about 20 or 12 hours. Because it starts with a fruity smell and the crumb gets sticky and wet, and then you see, like on the picture, that these ropes are formed. Therefore, these microorganisms are also called ropiness former. In the regard of fungi, the contamination mostly appears after the baking process. And yeah, the problem indeed with fungi is not only that they spoil the bread, but they also compose a health hazard to the consumers, because they can produce so-called mycotoxins, so secondary toxic metabolites, which are hazardous to the consumer. Therefore, within our lab, we asked ourselves, how is the potential of several isolates of lactic acid bacteria, isolates obtained from sourdough, regarding the potential to, on the one hand, inhibit the growth of fungi, but also afterwards inhibit the growth of bacilli. So first, we have tested several isolates of the genera or of the group Lactobacilli, the genera Pediococcus and Weissella, against the indicator fungi Aspergillus fumicatus, Aspergillus brasiliensis, Aspergillus flavus, Penicillium roqueforti, and Fusarium graminarum by the so-called dual culture overlay technique. And what we found out is that the antifungal activity was dependent not only on the isolate that was tested, but also on the, not only on the lab isolate that we tested, but also on the indicator fungi strain that we applied. Generally, the activity against the Fusarium graminarum fungi was highest because it was inhibited by approximately 94% of the isolates tested. And that was followed by the inhibition of Aspergillus fumicatus, which was inhibited or strongly inhibited by 55% of the tested isolates. Compared to that, the fungi Penicillium roqueforti showed the highest resistance, as only 6% of the tested isolates showed high inhibition of that fungi. And compared to that, 67% of the isolates did not show any inhibitory capacity against this fungi. But overall spoken, the isolates Lactibacillus plantarum S4.2 and Lentilactobacillus parabuchneri S2.9 showed the highest inhibition capacity against all the tested fungi. Indeed, it is always important to also have a look of that antifungal activity, not on an agar plate, but also within the matrix sourdough and bread itself. Unfortunately, we didn't have time yet to do it in our lab, but there is already an interesting work of Russo et al. Was performed in 2017. They prepared bread samples, on the one hand fermented with Saccharomyces cerevisiae, and on the other hand with a lab starter and Saccharomyces cerevisiae. And what they found out is that also the inhibition of Penicillium roqueforti was quite low or not at all present, but also the fungi Aspergillus flavus was highly inhibited by the starter culture, by the lab isolate, and also the species, a species of Fusarium was highly inhibited. So this result somehow can correlate to our results of the agar test plates. On the other hand, we have also tested the antibacillus activity. We again used isolates mostly from traditional Austrian sourdoughs and tested them against several bacillus species by the spot on the lawn technique. And what we again found out was that the inhibition capacity was again not only species dependent, but also strain dependent. And in general, the inhibition of Bacillus cereus was strongest compared to the inhibition of Bacillus subtilis as 15.20, which was already an isolate of the sourdough, so which was already adapted quite well to the environment of the sourdough. And Bacillus subtilis as 15.20 showed the lowest inhibition. Generally, the Lactibacillus plantarum and Lacticacibacillus paracasi showed the highest inhibition activity against the tested bacillus species, and Pediococcus pentasaceus showed actually the lowest capacity. Again, important to have also a correlation within bread and the matrix bread. And also in this case, there's already an interesting article of Paper et al. In 2003. And also they, on the one hand, artificially contaminated the bread with Bacillus subtilis, which was fermented by or with Saccharomyces cerevisiae, and on the other hand, they artificially contaminated the bread with Bacillus subtilis, which was fermented with Saccharomyces cerevisiae and also a lab strain. And also in this case, they saw a delay of the ropiness by the addition of the lab strain. Compared to that, the spoilage within the bread only fermented by Saccharomyces cerevisiae already showed signs of ropiness after 12 hours of incubation. So coming already to my conclusion, due to the lab work we have performed at BOKU, we could already see that the lactic acid bacteria, which were isolated from traditional Austrian sourdough, have antimicrobial activity. And also the results of other research papers, like, for example, Paper et al. And Russo et al. Showed that the fungal growth can be delayed, or the growth of bacilli can be delayed. That leads us to the fact that the sourdough processing has an effect on the shelf life and can maybe prolong the shelf life. What is also always important to have in mind is the staling of the bakery products, and also there in this issue research is going on, and it also seems like sourdough processing can delay the staling and so extend the freshness of the bakery products. But indeed, it is important to also view the interactions of present lactic acid bacteria with present yeasts, with process parameters that are used, and with the food matrix, because all these factors might modulate the antagonistic activity of specific selected lactic acid bacteria against fungal strains. So now I again have a live poll question for you, but it is again the same as at the beginning. Maybe you haven't changed your mind. Maybe some of you now think who were still irresolute that yes, due to the enhancement, the enhancement of the shelf life, it contributes to sustainability. Maybe less food, less bakery products are thrown away. So please again, I'm asking you for your vote. So I'm already curious if you have changed your mind, if still most of you say yes, if there's still some who are not certain. So I'm glad that I was able to somehow change the opinion of some of you. So more people do now think that sourdough processing contributes to sustainability of bakery products. I'm happy to hear that. And yeah, so I'd like to finish my presentation. So I'd like to thank you for your attention. I'd like to thank the ICC for giving me the opportunity to present you my work, and maybe help to to reduce food waste. And I'd also like to thank my supervisor, Konrad Domik, for his support, and my master student, Claudia Ammer, who performed the laboratory work. Thank you.
Thank you very much, Vera, for this excellent presentation with very surprising results from my point of view. Hopefully, we will discuss that later on. Now we come to the next speech. As I introduced to you at the beginning, Lisa Kall also worked in the course of her PhD thesis with sourdough. She focused on tolerability. Lisa, we are very curious and looking forward to your results and speech. Please.
Thank you. Thank you, Alfred. So as you mentioned, in this part, we will focus on the effects of sourdough processing on the tolerability of wheat. As you know, the consumption of wheat can relate to different diseases or different intolerances, and that already leads me to my first poll question. So we would like to know your estimation on the following question. So how many people are estimated to have an intolerance to wheat? Do you think the prevalence is below 1%, up to 5%, up to 15%, or up to 30%? Please let us know what you think. I'm very curious about the answers. I know that at least in some people around me, so friends and family, they really tend to actually overestimate the prevalence of wheat-related diseases. So I'm curious what you're thinking. Okay, so we really have a very wide range of estimations, and that is what I expected. And yeah, so I will go back to my presentation. Of course, the answer is somewhere in the middle. I'm afraid I can't really switch the slides. I'm sorry for that. No webinar without any technical issues. Okay, so now it's working. Okay, so here we are. So as I said, the answer is somewhere in the middle. We have to differentiate between 4 different wheat-related diseases. Only around 1% of the population is affected by celiac disease or wheat allergy, but the consequences of these diseases can be very dramatic due to anaphylactic shocks or villus atrophy. So it's very important to diagnose celiac disease and wheat allergy as soon as possible at a very early stage. And for the diagnosis, specific biomarkers have been identified, and the doctors can follow a very clear and validated procedure. And that is in contrast to the so-called hypersensitivities, including non-celiac wheat sensitivity and irritable bowel syndrome, or IBS. These hypersensitivities are in constant discussion because there are many unknown factors and there are no clear biomarkers for a diagnosis. And also there are no clinically validated procedure for the diagnosis. So people affected by these hypersensitivities are basically just identified by excluding the other diseases, which is also very tricky because all of these wheat-related diseases share a broad range of very similar symptoms, including the classical gastrointestinal symptoms, but also extraintestinal manifestations like headache or dermatitis, for instance. And that's also the reason why we don't have exact numbers for these hypersensitivities, but we assume that the prevalences are significantly higher than for wheat allergy and celiac disease. And in total, it is suspected that up to 15% of the population cannot tolerate wheat, or only in smaller amounts. So the triggers of these wheat-related diseases can be different cereal proteins, but also cereal carbohydrates, like the group of FODMAPs, which is a group of fermentable polysaccharides or oligosaccharides, disaccharides, monosaccharides, and polyols. And they have been identified as triggers of IBS. Among the cereal proteins, of course, gluten proteins are the main triggers of celiac disease and wheat allergy, but also amylase trypsin inhibitors, or ATIs, are often mentioned in connection to wheat-related diseases. And they have long been known as major triggers of Baker's asthma, which is a respiratory form of wheat allergy, but they also have been identified as triggers of non-celiac wheat sensitivity. One thing that is problematic about gluten proteins and ATI is that they are highly resistant to proteolysis. So they are not degraded or very poorly degraded in the human gut, and therefore they can very easily trigger inflammations or symptoms. In some cases, it's not totally clear what causes wheat-related diseases. Gluten proteins, of course, can also contribute to hypersensitivities, and also vice versa. There are some studies that claim that ATIs are involved in the mechanisms of celiac disease, for example. And besides ATIs, of course, there are also other non-gluten proteins that can trigger adverse reactions to wheat, but ATIs seem to be involved in all 4 pathologies, so that's why a lot of research is focused on this group. So we know that these wheat components or cereal components can trigger wheat-related diseases, but we are also interested to know what quantities can actually trigger symptoms. For celiac disease patients, it's recommended to totally avoid gluten in their diet. There's also a legal threshold level of 20 milligram per kilogram that is considered as gluten-free or as safe for celiac disease patients. There is, for ATIs, for FODMAPs, sorry, there is also, or the cutoff levels have been established by the working group of Jane Muir in Melbourne. And these cutoff levels relate to specific FODMAPs that are present in food. And the FODMAPs that are mainly present in cereals are fructans. So we have to keep this value in mind. Fructans are fructooligosaccharides, and we have to consider this value if we want to evaluate if a cereal product is can be considered as low FODMAP or high FODMAP. But we usually don't consume the wheat kernel as it is, but after a few processing steps. So we also have to consider the changes that occur during these processing steps. Fructans can be either degraded to fructose, or they can be converted to polyols, and both of them are also considered as FODMAPs. So we also have to keep these values in mind. In contrast to gluten and FODMAPs, there are no sound recommendations for an ATI reduction, so we need more clinical studies for that. So the aim of our study was to check whether sourdough processing can be used to improve the tolerability of wheat by degrading potential triggers. As you've heard in detail in Vera's presentation, sourdough is composed of loads of different lactic acid bacteria strains and yeast strains, and these microorganisms have special properties or enzymatic activities that can help us to degrade undesired wheat components. For instance, yeasts feature an inulinase or invertase activity, so they are capable of degrading fructans step by step. And this enzymatic activity is highest at a pH of around 4.5, which is usually not the case in yeast doughs. So the acidification that is triggered by lactic acid bacteria in sourdough can additionally help or additionally support the fructan degradation in sourdoughs by creating optimal pH conditions for the invertase and inulinase to work. Also, some of the lactic acid bacteria strains are capable to utilize fructose as a carbon source. So we can avoid, or the fructose that is released after the fructan degradation can already be utilized in as a carbon source, so we can avoid the accumulation of fructose, which might also be problematic for IBS patients. And besides these synergistic effects, protease, lactic acid bacteria also have protease or endopeptidase activity, so they help to degrade the potential triggers that are proteins. And that is what we also tried to investigate in our project. So we started with more fundamental experiments in model systems. We inoculated wheat suspension with different yeast strains that are usually common or very commonly present in traditional sourdoughs. And we quantified the fructan degradation by megazyme kits, and we also collected the fructan fingerprints by anion exchange chromatography, and we've seen that the fructan degradation ranged between 15 to 85%. So it was strongly strain dependent, and it was also structure dependent. We've seen that short chain fructans were more prone to degradation than long chain fructans. And the yeast that had the highest fructan degradation capacity was Saccharomyces cerevisiae, which was isolated from sourdough, which is basically the traditional baker's yeast. There were also other yeast strains with a very high fructan degradation capacity. They are impossible for me to pronounce. Charulsporo delbrueckii and Cluiveromyces marxianus. And some of these strains even had a very high fructose utilization capacity. So again, we can, after the degradation of fructans, they could utilize fructose as a carbon source and therefore thereby avoiding the accumulation of fructose. A very similar approach was used for ATI degradation. ATIs were extracted from wheat flour and then inoculated with different lactic acid bacteria strains. And here again, the ATI degradation was evaluated by HPLC and ELISA, and it was strongly strain dependent. We have ATI degradations from 50 to 85%. 85% was achieved by Pediococcus pentasaceus, but we are not sure if ATI degradation of 85% is sufficient to also reduce symptoms in ATI-sensitive patients. In a preliminary study, the lactic acid bacteria were grown on a gluten-based medium, and we've also seen that lactic acid bacteria were able to degrade gluten proteins, but the main aim of this study was the degradation of FODMAPs and ATIs. And of course, this degradation is also accompanied by a degradation of gluten, but we were not focusing on that because even very low amounts of gluten proteins are still highly toxic for celiac disease patients. So after these laboratory experiments, we also tried to evaluate the results in bakery trials. So we conducted, let's say, semi-industrial baking experience with our industry partners. And again, sourdough processing was found very effective in ATI and fructan degradation. Up to 80% of the ATIs were degraded, but again, we don't know if that is enough. And fructans were degraded by up to 90%. We also evaluated, I don't know if you see, sorry, the right slide right now. This slide is not working. I don't know what the troubles are. So it makes no sense at all what I'm telling you right now. If you don't see the right graphs. Okay, so now it's working. I'm sorry for that. So on the right-hand side, you see the baking trials for ATI and fructan degradation. We also try to evaluate the impact of the fermentation time. So the baking trials were conducted either by after a very short fermentation time of 1.5 hours or a long fermentation of 16 hours. And we saw that this had very low impact on the final ATI or fructan degradation. And we assume that most of the degradation takes place in the first hour of fermentation. We've also seen that the more sourdough starter we add, so the more cultures, the more lactic acid bacteria and yeast, the higher was the final degradation. And in another trial, we compared the efficiency of sourdough processing with the efficiency of yeast fermentation. And we've seen that sourdough was definitely more efficient in fructan degradation than yeast fermentation. Also, you see that by yeast fermentation, there was an accumulation of fructose that is released from the fructans that are degraded. And this, as I said, can also be problematic for IBS patients. So you can avoid that by either specifically selecting yeast strains that also have a high fructose utilization capacity, or as you've seen, by sourdough processing. However, I have to mention that even the yeast fermented bread can be considered as low FODMAP if we apply the low FODMAP cutoff levels that were established at the University of Melbourne. We also tried to compare the digestibility of sourdough fermented products to the digestibility of yeast fermented breads. So we simulated the digestion by using different enzymes and the different pH values at the temperature of 37 degrees. And we've seen that the digestibility of sourdough fermentation was increased in all 6 samples when we compare it to the yeast fermentation, at least in our in vitro experiment. So based on these results and based on also results from other working groups, there is a strong influence of yeast and lactic acid bacteria strains and counts on the degradation of potential triggers The degradation is also influenced by the processing conditions, so by fermentation time, for instance, but this impact is definitely lower than the impact of the sourdough cultures itself. But I think if we consider all these factors and these results, it might be possible to really specifically select sourdough cultures that have the desired properties that we want. But still, there is one question that is still open, or one question remaining, and that is whether a simple reduction of triggers or a degradation of triggers by sourdough processing is also enough for the reduction of symptoms in patients that are affected by wheat-related diseases. And there are only very few studies that really address this question. I collected some of them to present to you today, but I have to mention that most of these studies are based on in vitro experiments, so they are not actually conducted in patients. This is a study of Professor Chopin from Germany and Professor Gensler from Canada. And the approach is quite similar to the ones that we use. They extracted ATIs from wheat and then inoculated the ATI extract with different lactic acid bacteria strains. And after the fermentation, they determined the bioactivity of ATIs, so the inflammatory activity of them, because ATIs are capable of triggering specific receptors in the human body or cells of the immune system, and that causes the production of inflammatory markers or cytokines. And these cytokines can be quantified by immunoassays, and that's what they did. And they found a significant decrease in the production of inflammatory cytokines after fermentation of ATIs with lactic acid bacteria strains. So the inflammatory potential of ATIs was decreased after fermentation. The same working group also performed an in vivo trial in mice. You can see that this working group is actually very active in this area. And in this trial, mice were fed an ATI diet for 1 week. And the authors observed an barrier dysfunction and immune activation after this week. And in another trial, the ATIs in the diet were pre-incubated by lactic acid bacteria strains, or lactic acid bacteria strains were additionally added to the diet. And after this trial, the gut dysfunction was improved and the production of inflammatory cytokines or inflammatory markers was reduced when compared to the other studies where ATIs were not treated with lactic acid bacteria. So in this study, the authors were actually able to confirm the studies of the in vitro assay that I presented to you before. But still, this study is also, or the significance of this study is also somehow limited because it was conducted in mice and it was not conducted in clinical trials, including actual patients. The situation for FODMAPs is quite similar, actually. There are very few studies that actually evaluated the effect of sourdough processing in actual patients. Here we have 2 studies from Finland. They are based on dietary studies of actual IBS patients. So the patients were given either a sourdough fermented bread, which was low FODMAP, or a classical yeast fermented bread. And the patients that were given the sourdough fermented bread, they reported milder symptoms when compared to the other control group. But in this case, even the authors claim that the results have to be interpreted with caution because of the experimental setup and also because the evaluation of symptoms by questionnaires is not a very validated procedure. Still, the results has proven that sourdough is very capable of degrading potential triggers. The products, however, will still be toxic for celiac disease patients. But there are also some studies that reported an improved texture and an improved palatability of gluten-free products when sourdough processing is applied. So at least this might be one beneficial property of sourdough even for celiac disease patients. ATIs and FODMAPs were degraded very efficiently, but in this case, we are not sure if this degradation is also leads to a reduction of symptoms in actual patients. Even for healthy people, sourdough processing is often claimed to have beneficial effects. Some studies report a lower glycemic response due to sourdough processing, an improved mineral bioaccessibility, or an improved overall digestibility, as we've also seen in our in vitro experiments. But here again, we have no clinical studies or very few clinical studies to really evaluate the effects of sourdough processing in patients. But I think if we have this data, if we really have a basis of clinical studies, and then I think that sourdough processing is a very promising tool to degrade potential triggers, and it might also be very promising to for the development of cereal products that are especially useful for people that are affected by wheat-related diseases. Yeah, thank you for your attention, and I'm very curious about your questions.
Thank you, Lisa, for this very important and really impressive results. Thanks very a lot. I hope all the attendees agree upon that we don't finish our very interesting webinar at 4:30 because already we reached 4:33. But please, I ask for your understanding that maybe not all of your questions I see can be answered. But first of all, one person I have to point out, Fred Browns, your question number is impressing. Welcome to our webinar. Fred Browns is one of the team members in the whole grain working group of ICC. And so I welcome Fred, you especially. So, Fred, one of your first questions to the first speaker, to Vera, was a very interesting one, but I think a very complicated one. So, Vera, I have to warn you. I will try to read. You address better sustainability of sourdough related to one aspect of food waste caused by fungal spoilage. But starter backslopping making creates quite some starter waste, and prolonged fermentation time may have an energy related cost, etc., which are now not included. What would be the sustainability score if the entire product life cycle is looked at? Vera, a really complex question, because also energy intake aspects are in the question. What do you think about?
Oh, wow, thanks for that question. It's a really tough question. Yeah, it's a really good question actually, because we always need to take also into account of what it takes for the production actually. I guess there is no right answer. There's no yes or no in this case. But if I just think about the bread, if it's like not fermented for a long time, and if the industry like adds certain ingredients for increasing the shelf life or to increase flavor, that also needs the production actually. So that also has a cost effect. So I guess it would be interesting to have a deeper look into that subject and to then see, okay, from a production line, only from the production line, how is it then effective actually? That would be very nice to know. But yeah, I don't have, I guess, the answer right now.
There's already there's also a working group at BOKU that is focusing on sustainability of processes. So we might actually collect all the data we have for sourdough processing and ask them for their evaluation.
Okay, thank you. Thank you also for the additional comment. Again, a question coming from Fred Browns to Lisa. Lisa, are ATIs extracted from flour and exposed intact to cells in vitro or to animals structurally similar to ATIs that have been food processed by fermentation, heat processed by baking, and exposed to intestinal digestion? Do you expect differences in vivo responses in humans?
Okay, that is an excellent question. And there's no clear answer to that, because ATIs, as you know, Fred, it's a very recent topic. It's we don't have all the answers yet. We are still working on the molecular assessment of ATIs. So we actually don't know. There are some studies that say that the degradation, because ATIs can be present as monomers, dimers, and tetramers. And there are some studies that claim that the degradation of tetramers to dimers and monomers already has an effect on their bioactivity. So they claim that it is reduced, but again, these are in vitro studies, so we actually don't know, and we have to look into detail, even at this molecular level of ATIs. We have to perform immunological characterizations of ATIs. So of course there are a lot of differences when we extract ATIs. We try to extract them as pure as we can. So in our case, we extracted them by a mixture of chloroform methanol, and that of course changes the aggregation state of ATIs, which is not the case if we have ATIs in the cereal product.
Okay, thanks, Lisa, for this answered explanation. Vera, another question addressed to you. I hope we can clear something with this question. Vera, you mentioned GABA. So for me, it's the abbreviation for gamma aminobutyric acid to be a health promoting compound. The quantity in bread is so low that no effect can be expected.
So yes, the amount is quite low. It would be interesting if, so in general, not every lactic acid bacterial strain is producing GABA. And also always in different amounts. So what would be interesting to look into is, on the one hand, which strain does produce GABA, and on the other hand, also have a look on the amount that is produced. So yes, there is still the question mark, does then the GABA really have a health potential for humans actually? That's also something that's still interesting to look into. Each or every traditional sourdough is actually different, so we can't just say, okay, within sourdough there is always GABA present. It also is always strain dependent, so that's really important to point out here.
Thanks very much, Vera. Lisa, I dare to summarize two questions because they are referring to the same topic. You mentioned that accumulation of fructose would be problematic for IBS patients. Sourdough fermentation is known to increase free fructose content in bread and dough. So then this would be problematic. But I go on to the next question of Fred Brown's, because it's also with the focus on the digestion or degradation of fructans. I understood that in the presence of glucose, fructose is not the preferred fermentation sugar, explaining why free fructose accumulates as a result of fructan degradation. Would you agree?
Okay, so thanks for that question as well. Yeah, you're right. It's not actually free fructose, which might be problematic. It's actually excess fructose. So the amount of fructose that is accumulated when compared to the amount of glucose, which might be problematic. And that is also how it's claimed in the cutoff levels that were established by or at the University of Melbourne. So they really state the excess of fructose. And in our studies, we've seen, as when we determined the free fructose that I presented, it's always the free fructose that is in excess of glucose, and that was below the cutoff levels that were established. But even for sourdough, or even for yeast fermented bread as well. So actually that, might not be problematic in either case. And the second question, can you repeat that again, Alfred?
Yes. I understood that in the presence of glucose, fructose is not the preferred fermentation sugar, explaining why free fructose accumulates as a result of fructan digestion.
Yeah, I think I would agree to that, because I mean, if we degrade fructanes, which are usually composed mainly of fructose, and we and the yeasts utilize glucose in, or glucose, of course, is preferred by yeasts, then of course that is why the why fructose is accumulating compared to glucose.
Okay.
I would agree on that.
Thanks, Lisa. Yes, due to our time frame and it's 4:45, maybe I ask a last question, also coming from Fred Browns, again addressed to Lisa. Lisa, as you state, only specific proteases break down gluten and ATIs. Does this allow to claim that sourdough has this benefit in general?
Yeah, so we've seen that all of the strains were capable to degrade ATIs and gluten proteins, so that were the only proteins that we are looking into. Of course, the degradation of gluten proteins was definitely lower than compared to the degradation of ATI products, but we've seen a degradation in all of the strains. So yeah, of course, it's again, we investigated the product as it is. So we really investigated the outcomes of or the chemical outcomes of sourdough processing. We cannot conclude that it is therefore better tolerable, but it definitely affects the degradation of proteins. I think in general.
Okay, thank you very much.
If you would like to have a Zoom call soon, we would like to discuss all of the question again.
Okay, I think this would be a recommended progress in this discussion to come maybe to an end. Thanks very much to Vera. Thank you very much, Lisa. Really two highly interesting speeches with sometimes also surprising results. And as I promised all the attendees, we will hear very actual new results. And thanks for your interesting presentations. Thanks for your interesting answers. And at least I thank all attendees for participating this ICC webinar focused on sourdough. And I think the organizer, not the moderator, hopefully, will send further questions addressed to Fred Browns. We will try to answer all of your questions you asked. And maybe we could not only in a video conference, but also per email we could answer the residuing questions we had. Let me say thanks to you all, and hopefully we meet again in one of the next ICC webinars and training activities. Please watch all the data referring to webinars and to conferences on the ICC homepage. And at least I tell you all, stay healthy. And hopefully, if a lot of all around the world is vaccinated, maybe we are able not only to meet in video conferences, but also in presence talks again in ICC congresses. Thanks and goodbye.
Goodbye,
Bye.
everyone. Thanks for listening.